Welcome to Thermo Fisher Scientific Corporate Satellite Symposium at the European Society of Human Genetics 2020 annual meeting, virtual meeting. Today, we will focus on how human genetic research can pave the way to bring precision medicine into the clinic. Speaking is Luca Quagliata, Global Head of Medical Affairs for Thermo Fisher Scientific. As human genetic research is essential to develop new paradigms in healthcare, we asked our speakers for today to illustrate with their talks how this is really happening. And by doing that, we will cover two broad themes. At first, how genetics can help to identify individuals at high risk of disease, to focus screening and early intervention where they can have most impact. On the other hand, we will talk about how genetics can support improved diagnosis and treatment for better health outcomes in patients. Now, a very current example of these themes is COVID-19 pandemic. Multidisciplinary research has accelerated very quickly these days to characterize the virus and its epidemiology, to improve detection and diagnosis of the virus, to develop vaccines and other treatments, and to understand the genetics behind susceptibility, severity, and treatment response. Leveraging on our unmatched portfolio of instrumentation at Thermo Fisher Scientific, we have now worked urgently to develop new solutions for the COVID-19 research across this workflow. Please take the chance to find out more information about this visiting the What's New page on our company corner. But now, to set the stage of our symposium, let me first focus for a moment on one thing, that is risk stratification. Risk stratification is needed not just in infectious disease, but across all common disease, of course. Risk stratification today means enabling a paradigm shift from single gene testing used in rare disorder to the analysis of complex variants across the entire genome. Because, for example, polygenic risk scores are now gaining momentum as a valuable approach to profile risk, because while they consider each of the many variants, they also allow to estimate the cumulative risks that they contribute altogether. Example where clinical benefit of polygenic risk scores has been proposed include improving CAD, cardiac artery disease risk estimates to define individual most likely to benefit from statin therapy, or targeting prostate cancer screening to population at high risk. Later in this symposium, Dr. Richard Fidham from Cytox will speak about application of polygenic risk score to Alzheimer's disease risk profiling using the Applied Biosystem Axion genotyping platform. Cytox developed a custom array with us, but arrays such as the Axion Precision Medicine Diversity Research Assay also provide excellent tool for polygenic risk score research. Deep genetic insight can be derived from over 850,000 SNP markers, which can be customized to your study. The array design includes genome-wide coverage, extensive pharmacogenomic markers, pathogenetic variants, and disease risk markers, blood typing markers, and other content. Once again, please explore our company corner to learn more about it. A second topic that I would like to touch upon is the need to get faster and comprehensive insight about all relevant disease genomic drivers to make the most informed and appropriate treatment decision. But what that future could look like? In this slide, you see, for instance, the impact in the context of cancer patient management and therapy administration. But in general, for cases where time really matters, like cancer, phenylketonuria, cystic fibrosis, or severe combined immunodeficiency, just to name a few. Having comprehensive information about the underlying genomic drivers and thus a better overall disease profile can help to improve outcomes. Therefore, the research priority nowadays is to find a path to NGS protocols that are as fast as possible and as simple as possible while delivering accurate results, no matter the setting in which they are generated. And now, I'm delighted to introduce to you the Iontouring Genexis system to power this research effort. This is the first turnkey automated NGS solution, which enables in-house NGS accessible testing like never before. It features a single-day turnaround time, a specimen-to-wrapper automation with only 2 user touchpoints. It allows analyze individual sample cost in a very cost-effective manner. And notably, the system and its consumable are manufactured at facilities that are registered with the FDA and under ISO 13484 certified. Now, research application on the Iontouring Genexis system span from oncology, inherited and infectious disease, with reproductive health as a near to come in the future application. The Iontouring Genexis work environment comprising of samples extraction step, a fast system that will allow sample prep within a 2 hours turnaround time, and the actual integrated sequencer. With a brand new design Iontouring chip, working at the speed that you've never seen before. To run a single lane, it will take you as low as 14 hours and approximately 24 to 13 hours for the full chip, meaning from samples to report within just about a day. And now, if you'd like to know more and have more information about what I showed to you in those slides, please complete our form to be contacted by one of our representatives. Now, it is my pleasure to introduce to you our speaker for today. At first, we will have Dr. José Luis Costa, an affiliated researcher from the Ipatimob in Portugal. He will be talking about custom Iontouring AppleSeq panel in combination with the Iontouring Genexis system for rapid RASopty testing. So he's going to share with us the result of his pilot study. We will then move on with Professor Carl Goodyear, who is currently working at the University of Glasgow in UK. He will share with us some of these recent results about molecular profiling of autoimmune inflammatory disease. And at last, we will have Dr. Richard Piner, CEO of Cytoke, based in Oxford, UK. Dr. Richard Piner will share with us some of the latest data that they gathered around polygenic risk score assessment in the context of Alzheimer's disease, with the aim of transforming early detection and clinical management for this patient. And now, with no further delay, I would like to ask Dr. José Luis Costa to start with this presentation. Please enjoy the meeting.